Hi everyone, welcome to the Angling Trust Fisheries Forum tonight. My name is Richard Pittman and I shall be hosting a presentation all about the Environment Agency's National Coarsefish Rearing Unit in Calverton. The idea behind this presentation is to give everybody an insight into the processes involved in rearing such a large quantity of coarsefish. These fish are then stocked out nationally across the country into waterways the length and breadth of England. In this clip here, we can see the size of the farm and how it's split up into different types of ponds. These ponds across here and here are our second year growing on ponds. All the ponds inside the greenhouses are our larvae ponds, including these smaller three here, which are used for dace. And then you can see the scale across the road. There are 4 large on-growing ponds where we grow our fish for the second summer. In these we can have up to 60,000 fish per pond, varying up to 5 different species. The majority of the fish that we stock out across the country come from this side of the road. The farm is funded solely through fishing licence income, and we supply each area on this map annually. The star in the centre dictates our location in the country, which means it's only a short journey to all of the major travel routes, so we can head in any direction. We produce 9 different species of coarsefish here at the farm, some of them riverine, some of them solely still water, and each of them has a slightly subtly different requirement from the next. Annually, we stock out up to 500,000 fish across the length and breadth of the country, mainly into rivers, but obviously into still waters too. All the broodstock used on the farm are wild fish. We collect them from places in the wild where fish would naturally be gathering to spawn at that time of year. Using wild fish instead of captive fish means different parents every single year. This ensures a wide genetic range throughout the species that are going to be stocked out throughout the country. The adult fish, as I say, are collected at a time of year when they've done 90% of the work for us. They're in spawning condition, they're ready, and we go along, break up the party and borrow the fish. Here's a beautiful picture of a roach, male roach in spawning conditions. You can see the tubercles on its head. There we go, get the pointer up. You can see these tubercles on its head. This is the condition that we collect these fish in. Again, another beautiful picture of barbel spawning. And if we look at the fish at the centre, this is the larger female. And there's a male on this side and a male actually on the other side. You can see its other fin there. So she will be releasing her eggs and the males will be releasing their milk. These are the places we target, as we know that the fish will be there in abundance. We use electric fishing to collect the adults, plus other ways of collecting, rod and line, netting, fike nets, depending on the species. Only a very, very small current is put through the water. We're only fishing maximum of 1 amp. We have to remember that the fish that we're targeting are very, very delicate at this time of year. The eggs are full of the females are full of eggs, sorry. The males are producing milk, and the last thing we want to do is cause any damage to these fish. They don't belong to us. We borrow them from angling clubs. So we have a short clip here of electric fishing taking place. You can see how the fish are there in great abundance, meaning we get to widen the genetic variation from the adults. There's no point collecting them from areas where there's only a few fish, as you wouldn't get the diversity. The fish are only stunned for a short period of time and then put into recovery in a tub full of oxygen, so they recover extremely quickly. You can see how the electricity isn't holding them as it does with some electric fishing processes. We're just turning them over. We're just tickling them, being able to catch the fish that we need, and then the rest recover and carry on with their business. There's small and large fish mixed together in these sections, as the places we target, especially on the river, are the natural spawning grounds. As I say, the places where fish come to spawn naturally. In this clip you can see how the chub aren't completely under. They're just being held just long enough for us to be able to net them, stick them in the tub, and then they recover. Once the adults have been selected, they're then loaded onto our specialist fish transport vehicles and transported back to the fish farm, into the hatchery. The vehicles have got tanks on the back, purposely built, with oxygen monitoring system, so at all times we can monitor the welfare of the fish. If for some reason the dissolved oxygen in the tank starts to drop, we can respond to that immediately. There's a number of compounds we use in the water, some which reduce stress and others which replace any mucus that may have been lost through the handling process. Once inside the hatchery, we have a number of individual discrete systems that the fish are kept in. These allow us to keep batches of fish completely separate from others. We may have fishing from the Trent catchment, we may have fishing from somewhere up north and down south, and we obviously don't want any of these to mix. All the effluent from here goes through a large ozone disinfection unit, meaning that no parasites from any other catchment could end up in ours. Inside here, we can control the day length, we can control the temperature, so we recreate all the natural conditions that the fish were collected from, so there's no shock as they're brought into our hatchery. The males and the females are kept within the same system, but not directly in the same tanks. This means that spontaneous spawning can't take place, which means we wouldn't be able to harvest and collect the eggs. Obviously, we've already sexed these fish on the bank, and in this slide you can see how easy it is to sex certain species. You can see there, this is the female barbel with quite a large protruding papilla, and the male has a fairly small one, and he's already producing milk. With species like barbel, it's very, very easy. If we move on to some of the other species we produce at the fish farm, say grayling, we use other characteristics to help us with the sexing of the fish. With grayling, the colour is often different between the males and the females. The males are much darker, as depicted in the picture on the left. We can also use body shape as a cue. The female is generally more plump, more rounded, as she has the egg mass inside her, whereas the male is still quite slim. We can, if we want to, take a small sample of eggs from a female and see how far along in the spawning process she is. What we do is we clear the egg. The last piece of the egg to clear is the nucleus, and from that we can tell how close to ovulation the female is. If the nucleus is still slap bang in the centre, she's a way off. The closer the nucleus is to the edge of the egg, to the hole where the sperm goes in, the closer she is to ovulation. The next stage of the process is to administer a natural hormone dose to each of the fish. What this does is bypasses the fish's own natural production of the hormone and gives us a degree of control over when the fish will ovulate. This ensures that we can collect the eggs, and it also minimises the amount of handling needed for the individual fish. The eggs inside the fish ripen further, the nucleus hits the side of the egg and breaks down, and at this point the fish is now ready to ovulate. In this slide we can see the difference between prior to hormone injection and post hormone. In the prior, you can see how the egg mass is still quite high up in the fish. The eggs are still held within the lamellae. They're hydrated, but they've not been released. There's still a little bit of hydration left to go. Once the injection's been given, you can see how the egg mass has slumped down into the body cavity. This fish has released all her eggs, and they're ready now to be stripped from the female. At this point now, the fish are very, very delicate. The eggs have been released inside the body cavity, and with slight manipulation, you can start to see those appear at the vent. The fish's abdomen feels extremely soft and almost jelly-like to the touch. Whenever we handle fish inside the hatchery, we make sure they are anaesthetised. This obviously minimises the stress to the fish and aids the handling, as some of the fish that we use are quite large and extremely strong. Once the fish is anaesthetised, the female is turned upside down or the right way, so the vent is pointing downwards, and the eggs are gently massaged out of the fish's body cavity. When stripping, we'll put one female in a bowl and 2 or 3 different males milked over the top. This ensures a wide variety of genetics in that one bowl. Also, if the milk from the male is no good, you haven't wasted a full ovulation. All the fish then go into recovery, where they recover fully and go back into the tanks. And from there, they can be returned to their natural homes in the river. Some of the fish, as you've probably seen in the pictures, have small blue dots indicating their weight on the underside of the fish. And we see these fish year on year in the river. We know that they have gone back to their natural homes, carried on successfully, and we can leave those fish in the wild and choose separate ones next year. This is a short clip of a fish, a barbel, being stripped of its eggs. Alan is applying gentle pressure to the fish's abdomen, and the eggs are running out down the anal fin and into the bowl. You can see this is the egg mass that's come out of the female. There are a good number of eggs in there. It's really only gentle pressure, and with the fish being anaesthetised, you can see that it doesn't move during this process. Here we have the male all being dried off, so there's no water that enters the bowl, so as not to activate the eggs or the milk. And you can see again, there's one male going over the top. This process then would have been repeated with another male, maybe 2, and then the eggs would have gone for fertilisation. Cyprinid eggs are extremely small when compared to salmonid eggs, so the eggs of a trout or a salmon. Barbel produce the largest of the cyprinid eggs that we deal with, and tench produce the smallest. When you talk about the amount of eggs a fish produces, the fecundity, tench can be anywhere up to 250,000 eggs per kilo body weight, whereas barbel can be a lot less. The eggs vary in colour depending on the species and the natural spawning substrate they would attach to. Generally, weed spawning fish produce a slightly more green egg so that it camouflages when it sticks to whatever weed it's going to spawn on. Gravel spawners tend to have more of a yellowy, orangey sort of egg, again to allow for that egg to be camouflaged once it gets into the substrate. Once the eggs and sperm have been collected, both in a dry state, they're passed through into the incubation part of the hatchery, where fertilisation takes place. In the wild, sperm is only motile for a very short space of time. With our fertilisation solution, we can extend the motility, the movement of the sperm, up to maybe 60 seconds, which maximises the number of eggs that are going to be viable. The eggs are then stirred very gently using a feather to aid the fertilisation and to keep them moving. Some eggs are sticky, some eggs aren't. The movement stops these eggs sticking together in one large clump. As the fertilisation takes place, the eggs begin to swell and take on the liquid that's around them in the bowl. We then wash the eggs to remove any spent milk, to remove any ovarian fluid and anything else that may be in there, so they're a nice clean bowl of eggs ready to be laid down to incubate. The eggs are then laid out into our troughs with a lovely constant clean flowing water supply to make sure there's enough oxygen distributed around the egg during development. They're laid down onto different types of substrate depending on the eggs. During the incubation, the sticky eggs are spread out onto a filamentous kind of substrate. It's a geotextile. This separates the eggs, keeps them apart from each other so you don't have any big clumps, as the ones in the middle wouldn't get the water supply, the oxygen supply, and they would perish. Non-sticky eggs, species like barbel, grayling, they're laid down into trays without the filamentous substrate. These can then get their water flow all around as they incubate. This slide here shows the different stages of egg development just after fertilisation and through until we start to see the larvae form around the yolk sac. This is the first cleavage as the cell splits for the first time. Then second stage where it splits again, so we've gone from 2 to 4 and so on, right the way through till we have the larvae forming in this last one around the yolk sac. We also have a video which shows the egg development stages going through. Time-lapse video taken here by the staff members. You can see how the egg has gone through its different stages of formation. Then we start to see the larvae form around the outside and with the individual muscle blocks. And now the larvae moves around inside the egg. In a moment it will pause and we can actually see the larvae as it's developing. Here we can see the large eye, which has no pigment, big yolk sac, and the larvae's body as it's curled around inside the egg. And if you look closely, you can see individual blood cells travelling over the yolk sac, picking up some goodness, and then travelling around the fish's body. More development takes place and more movement inside the egg. The fish is active before we start to see pigment in the eyes. Again, in a moment it will pause, and now you can start to see actual pigment on the fish's body. We can still see that yolk sac, but it's got a lot smaller. We have the large eyes which now have pigment. This fish can now see, and we can still see the blood travelling over the yolk sac and even down around the edge of the fish's tail and back up the other side. This fish is now very close to hatching. A little bit more movement and it's gone. Once hatched, the larvae still continue to develop on the bottom of the trough. They're completely reliant on their yolk sac at this stage. The mouth parts aren't fully formed. They still have one large elongated fin, and they also don't have their swim bladder inflated. At this point they're extremely vulnerable. Everything in the wild wants to eat them. We produce their first food on site, which is Artemia or brine shrimp. Because it's a live natural food for the larvae, it triggers the snap response, that inbuilt hunting instinct that all fish possess. It's also full of natural enzymes and goodness that the fish need to kickstart the gut to work properly. The larvae grow really quickly on the Artemia. Such a highly nutritious diet. They then start to develop the fins and start to look a little bit more like the fish that we recognise. A high percentage of the larvae that we produce at the fish farm are returned back to the natural environment where they came from, the stretches where the mums and dads were collected from. And this stops any recruitment issues that could be caused by us borrowing a small percentage of that spawning population. While all that's been happening inside the hatchery, the spawning and the incubation, out on the farm, the ponds have been being prepared, ready to accept the larvae that are coming through. The ponds are now full, and what we're looking for is an abundance of natural food. What we want to see in there is we want to see the rotifer. This one is non-predatory. It's a great first food for the larvae, and again full of goodness which the larvae will grow very, very quickly on. What we don't really want to see in there are the copepods. These can be predatory, and if the larvae is still small, the copepod will take chunks out of them and even consume the whole larvae. The ideal situation is plenty of rotifer and maybe a couple of copepods. And as the stages go through, the fish grow quickly on the rotifer, and as the copepods are there, they're then able to consume those too. Once the pond is ready to accept fish, the larvae are introduced into the ponds. We stock up to 30,000 larvae per greenhouse pond, and the fish grow rapidly on the abundance of live natural food in there. In just 3-4 weeks, they can treble in size. Again, while all this is taking place, our outdoor ponds are being filled, worked, and treated in the same manner as the larvae ponds. What we're going to do then is we're going to stock out last year's larvae for their second summer of growth. As mentioned originally at the beginning of the talk, we have 4 large on-growing ponds as part of the farm, where we can stock up to 60,000 fish per pond. We use a technique called polyculture, where you stock different layers of your pond with different species. You utilise the bottom with maybe barbel, the midwater with, say, roach, and then the surface layer with a species like dace. Utilising every level of the pond ensures maximum growth for all species. Not only do we produce a lot of natural food in our larvae ponds, it's the same process for the outdoors. As you can see in this video, all of these little dots that you can see on the screen swimming around are daphnia. It's all natural food for the larvae to feed, for the fish to feed on. The fish that you just saw going in were barbel that had just had their first birthday, and they were going into a pond to grow on for the next summer. The amount of food in this pond is so abundant, no matter where the fish looked, there's something for it to eat, and that's incredibly important in that first few weeks after stocking. The daphnia don't swim in jagged lines, they swim in nice small circles, allowing the fish to hunt them down and consume them very, very easily. In the outdoor ponds we use aerators that not only oxygenate the pond but also supply flowing water for the fish that are in there. We're very aware that the fish that we breed mainly are riverine species, so we have to ensure that they build up the right muscle to be able to cope with flowing conditions. We want them to build up their red muscle. This is done by strategically placing the aerators in positions in the pond where if the fish want to feed, they've got to swim. The fish that we produce want to eat all summer long, so spend the whole of the summer in the pond swimming against these flowing conditions. Making sure the fish that we stock out into the wild is fit for purpose is something that we're very, very keen on doing. Otherwise, they don't have a chance of surviving. At the end of the summer comes the harvest period. We harvest the ponds through various methods, either draining or netting, depending on the size of the fish. And the fish are transported back to clean holding tanks with nice flowing water. Again, flowing conditions, so the fish still has to remain swimming and build up that red muscle that's so desperately needed for when it goes out into the wild. The sizes of the harvested fish vary between species and year class. Here in this picture we can see a crucian that's probably 8 inches long, right down to a barbel that's a first year fish, a couple of inches long. The fish that we produce here are never sold or never delivered by size. Naturally, in the wild environment, you get a varying size range between a year class. If you produce fish and stock them out by size, you could inadvertently be selecting out males and females for different sites, and this obviously isn't going to work. The fish we supply are a full range of the sizes, ensuring that there is no chance of that happening. Once harvested, the fish have to be processed. As we mentioned, we could be having 5 different species in a pond for polyculture. Those have to be hand split, as there's no machine to do that. Hand split, counted, processed into individual species tanks, ready to then be delivered nationally. Once processed, these fish can then be counted back onto a vehicle and delivered across the length and breadth of the country into varying different types of waterways. In this video we have a short clip of the counting and delivery process. The fish are being selected for individual species, put onto the counting table and counted through the fish counter. Here we can check to make sure all the fish that are leaving the site are of the species required. The fish are counted through there at quite a fast rate into a net and being able to be loaded up onto the transport vehicle. As we can see, the transport vehicle has tanks on the back specially designed to take these fish, baffles on the lid to stop water movement, and a beautiful oxygen supply in each tank, which, as I say, is monitored throughout the journey. We then drive to wherever in the country they've been requested, where we can offload them and release them into the wild. This is a video of some barbel going into the river in Yorkshire. This is the river wharf. And as we'll see in a moment, once the fish are released, they have no problem coping with the flowing conditions. The fish go in, they go down to the bottom, and a number of them actively swim upstream against the flowing current. This is because we've ensured that all the way through the process, from hatching to delivery, they've had the flowing conditions to be able to cope with these sort of environments. Success for us is exactly what you see in front of you. All these fish originated at Calverton, were stocked out into a wild environment, and have thrived and grown on to what you see before you. The grayling that was stocked earlier this year into a section of river where they weren't being caught, and now they're starting to catch these young grayling. What we don't want to do is we don't want to keep stocking places year after year if we know it's not working. For us, seeing natural recruitment coming through in the wild is what we class as a success. Populations are monitored by the area teams, and we can see whether the programs that are being undertaken are actually succeeding. I hope you've all enjoyed tonight's presentation. Thank you for watching and listening to me. Now you have a little bit of an understanding of what processes take place on the farm to produce the fish that we do. Please remember that we have a Twitter and Instagram account that you can follow, which will let you see more of the interesting work that we do here on the fish farm. Thank you very much.